Both inside and out, our bodies contain a number of microorganisms. Microbiomes are small and large colonies of viruses, fungi, bacteria, and other microbes, including viruses that attack bacteria. A study from 2016 states that microbial cells and human cells coexist somewhere around 1.3 to 1 ratio, suggesting they only slightly outnumber our own cells. So hang on, aren't they supposed to be dangerous? Well, it's a bit of spectrum. Some are pathogens and only harmful when they are placed in a wrong place. And some are very beneficial to the humans, such as they increase development of immunity, defense against pathogens, host nutrition, including production of short chain fatty acids, which are important in host energy metabolism, and they also influence on human behavior. So we have different microbes in different part of our body. For example, bacteria in our gut is different as bacteria is on our skin. But what about the microbiome on the things we own? For example, are microbes the same on my shoes as on my phone? To answer this question, I'm gonna do hypothesis testing. So our null hypothesis is there is no microbiome difference in samples collected from gadgets. An alternative hypothesis would be there is a microbiome difference in samples collected from gadgets. To run this experiment, Western Sydney University Hawkesbury Institute for Environment collected and sequenced data from different gadgets. This dataset was in gigabytes, and the information about this dataset can be found in a metadata file, which is a file that contains technical information. For example, we have different samples and we have collected samples from different people's phone, shoes, wallet, glasses, etc. And our job as a data scientist is to visualize all this sequence data so that microbiologists can easily understand nature of different microbiomes on different gadgets. They also provided a classifier for assigning taxonomy to our sequences. Now, to run this experiment, we will be using QIIME2 software, which is open source bioinformatics pipeline for performing microbiome analysis from raw DNA sequencing. QIIME is designed to take users from raw sequencing data generated on different platforms and process that data for quality graphics and statistics. This includes demultiplexing, quality filtering, OTU picking, taxonomic assignment, phylogenetic reconstruction, and diversity analysis and visualization. This software requires high amount of RAM and CPU power, so I will be running this experiment on Western Sydney University servers. This software generates two outputs. First one is result of analysis, which is also called artifact with suffix .qza, and a visualization with suffix .qzv. In the first step of QIIME pipeline, we do demultiplexing, which allows large numbers of libraries to be pooled and sequenced simultaneously during a single run on a high throughput instrument. Now let's see the visualization after demultiplexing. Now here I am on view.qiime2.org website. I have uploaded my QZV file after demultiplexing. Here is the basic statistic for our sample. But the interesting part is inside this interactive quality plot. You can see we have forward reads and reverse reads. With the help of this plot, we can take only those samples which have high quality. So this y-axis represent the quality from 0 to 40. So 40 is the highest as 100%. So if I click and drag this area in forward reads, it will zoom in and show me the quality of these samples. As you can see, the quality is dropping gradually, and at the end it dropped to very low number, and we have missing samples in this area. So you can see we have missing samples in this part from 40 to 63. So I will take only those samples which has highest quality, which are starting from 63 up to, let's say, 188. The reason why I'm not moving after this 188 part, because it started dropping the quality and it almost reached the plateau from 195 to 205, and after that you can see it's dropping up to 0. Now same for the reverse reads. The quality data is starting from the 11th position and it goes up to around 160. Now if I double click and hold my mouse and zoom this area, you can see that around 155 it reached the plateau, and after that its quality has started decreasing. And remember, this y-axis represent the quality of our samples. So I will choose from 11 up to this position 161. So this 161 has the quality around 30. So it's better for us to choose this 161 as compared to this 180 position, which almost has y-axis quality between 25 to 30. As discussed on the visualization, for quality control and feature table construction, I'll be using a plugin Dada2, which will have 63 to 188 bases for forward visual and 11 to 161 for reverse visual. The result of this method will create a feature table which contains number of frequencies of each unique sequence in each sample in the dataset, and a feature data artifact which maps feature identifiers in the feature table to the sequences they represent, often refers as OTU, operational taxonomic unit, which can be defined as an operation definition of a sequence data point that explicitly models the errors of the sequencing technology. Now let's see the visual for that. So here on QIIME website, when I uploaded my pipeline output QZV file, here are the basic statistics. We have 20 different samples. Now the interesting part is inside this frequency per sample. Now this median and maximum value defines the input for next QIIME pipeline. So if I show you this interactive sample detail, you can see we have this bar plot. So we have selected this item type, and here you can see different sample representation for glasses, for phone, for shoes. We can increase or decrease the number of samples by here. So we have less samples for water bottles and shoes. If I change it to show me the bar plot person wise, I have more samples of Michael gadgets as compared to Muhammad and Sui. And also if I change it to biological, we have more samples collected from males as compared to female. And for ethnicity, so we have collected more samples from Australia as compared to Middle East. Now the second output of this process is this file, which have name of the operational taxonomic unit and the OTU itself. So if I click here, it will take me to this US BLAST website where I can find more information about this particular OTU. Now the next step is to construct our phylogenetic tree. It helps us find relationship between OTUs and different samples, as the number of sequences are too many for humans to analyze and interpret. So instead, we can use phylogenetic information to get some enhanced diversity statistics that are embedded in a phylogenetic framework. Now, after running the pipeline, I am on this website ITOL, which is Interactive Tree of Life, and I uploaded my rooted tree file here. So this is the tree which we got. It has different length of branches and each branch has a sequence associated with it. As I'm not a microbiologist by myself, so I cannot interpret this tree. However, drawing this tree and showing it to microbiologists would be a great favor to them. Now, this phylogenetic tree construction process also returned one bar plot file which I uploaded on QIIME2 website. Here are some information according to different levels. For example, if I go to level 10, these are the bacteria which I find on level 10. You can see the name of these bacteria here. I can also sort these bacteria by item type. For example, these bacteria belong to this item of glasses. I can increase more levels to see more diverse information over here. Now moving to the last part of this video, finding diversity. First we have alpha diversity, which is the diversity within the sample. Here are a couple of methods: Shannon's observed OTU and Faith's phylogenetic diversity. Now after running diversity experiment in QIIME, this is the alpha reflection result. We have taken total 1636 samples for the first experiment of alpha reflection. The reason is, if I show you real quick, our older step where we removed the noise, this statistic table, you can see we have the maximum frequency of 1636 and the median was 659. So in the first alpha refraction curve, I'm mapping all of my samples. So first of all, this is our Faith PD metric and you can see different sample OTUs here. So this line basically show the operational taxonomic unit which we got after removing the noise. You can see that wallet has more samples as compared to water bottles, and if I change the metadata column to person, Jonas has more samples as compared to soy, which can be seen by this green OTU line, and Muhammad, which has very low samples, around 600 or something. So if I select this observed OTUs, we need to do a cutoff because some samples don't have enough coverage. So if we sample more, we will start losing. So instead of taking the whole sample frequency, I will make it a bit more lower. So here is another alpha refraction with 900 samples, and you can still see that although these three are increasing, Michael, Jonas and Jane, but we still have low convergence for Muhammad and Soy. So I created another alpha refraction to do the cutoff, and here is the last alpha refraction which has 650 frequency per sample, which was also our median in quality table. So as you can see that on this point of around 650, we have data from all of the people. So I will do a cutoff in our original 1636 frequency per sample at the position of 650 around here. So this is our selected alpha refraction after doing the cutoff. Now, as I said before, alpha refraction is checking the diversity within the sample. So as this curve is going up, it represents the data which we have is very much diverse as far as individual sample concerned. Now the next step is to find the beta diversity, which is finding diversity between samples. So I'll be using Bray-Curtis distance, unweighted unifrac distance and weighted unifrac distance to represent diversity within different samples. Now after running the QIIME pipeline, here I'm on QIIME website and I have uploaded my QZV file here. You can see that we have calculated diversity between different samples. So if I estimate the diversity using a p-value between Australia and Middle East, you can see we have a very large p-value. So our null hypothesis can be accepted here that there is no microbiome difference in gadgets from different regions because the p-value is very large. Now here is another visualization. This is a 3D visualization. We can see that Muhammad microbiomes are very much different to the rest of the people. Here is another 3D visualization using Bray-Curtis method, and this also proves that microbiomes on Muhammad's gadgets are very much different or far from other people. We can also select different attributes from this tab to check other hypotheses. Thank you for watching this video. I would love to have your feedback.